Good evening, everyone, and thank you for joining us. This evening, it's my great pleasure to introduce Dr. Shannon Rupert. She is the director of MDRS, which is the Mars Desert Research Station in Utah. And she is going to be speaking about, is there life out there? Current biological research at the Mars Desert Research Station. Again, MDRS. Shannon, take it away.
Good evening, everyone. Thanks for joining us. Okay, so I should probably start by saying that I am at MDRS right now. So that means I'm on satellite internet. So I'm probably going to lag a little bit. Seems to be slower in the evenings, but at least there's no wind. So tonight I wanted to talk a little bit about some of the biology work that we're doing out at the station that is not crew related. So we talk all the time about what the crews do, and they do such amazing work, and we share that with you. But we seldom talk about the things that we do that are non-crew related. And so I asked a few of my good friends to join me tonight, and we're going to talk about 4 different projects that we are doing at MDRS. So this is my backyard. And as you might imagine, as a biologist, as a field biologist, I have all this terrain to wander over, and it seems kind of a waste that I'm here all the time. And I don't have much research of my own. I did spend 3 years mapping dinosaur bones. That was really, really fun. But since I'm a biologist, I would like to stay within my field. So we have 4 current projects we're going to talk about tonight. The first project we're going to talk about is the work that we're doing in the Green Hab on the design and implementation of a traditional agricultural system for Mars. And I'll share a little bit of that research. And then, like I said, I have 3 other people joining me. Atila, who's our assistant director, also was an intern and then became a formal intern and was working on a project on microbial ecology right before COVID hit. So he couldn't come to MDRS and do the work, but he wanted to do the work, and he kept at it and he kept at it. And the result of that is our latest project, which is this geomicrobial mapping and characterization of discrete, visually identifiable, lithologically based communities at MDRS. Yes, Penny came up with that title. It's kind of a placeholder, but basically what we're doing is we're looking at patterns and growth in desert varnish and lichen, and kind of seeing who's where and who's winning in the place where they are. We're very lucky we have 3 interns coming this winter to work with us on this project. And then our longest, Paul Sokoloff is going to talk about our longest running non-crew project, which is the Martian Biology Project, which we started, I don't know, we probably first came up with it in 2017. And this is every year we meet out at MDRS and we do biodiversity surveys. They're not Mars related. They're basically just a really, really good opportunity for us to contribute to the science in this area. And then the last project we're going to talk about is the NASA Spaceward Bound Utah project, which is a NASA grant funded project that brings teachers to MDRS. We're going to have our 3rd cohort of teachers in about 2 weeks. And I'm super excited. This is an amazing program. The teachers are unbelievable. And I mean that in a good way. So let's talk a little bit first about this idea that we had to utilize our Green Hab in a way that will contribute not only to a more robust crop production system here, but also allow us to look into what people are saying about what we're going to do for farming on Mars, and try to put some of that into our program. Little background about greenhouses at MDRS. If you look at the upper left hand, that is the original greenhouse, that little round cylinder. A windstorm came up on Crew 4, blew it across the horizon, and we never saw it again. The next greenhouse was the one on the lower left. That's the one that most of you are probably familiar with. That's the Gary Fisher greenhab, and it was built to be a closed loop water recycling system for the station and a greenhouse. Unfortunately, it was too small for the load of 6 people when we included the kitchen gray water, which we did. I wish we would have had an opportunity to alter the system a little bit and see if it would have worked. But it became so overwhelmingly lived in by E. coli that we couldn't even grow crops in it. Finally, we retrofitted it as a greenhouse. We started growing crops in it, and unfortunately, it burned the ground. So you might remember we crowd-funded a new greenhab, and we built what you can see up at the upper right hand of this slide. And it was a geodesic dome. Once they built it, they realized there was no way to actually keep it wind and waterproof. And so being crowdfunded, we didn't want to tear it down, and you'll recognize it now as our science dome, and which I love that building, everybody knows that. And as a result of turning it into a science dome, we had to build a new greenhouse. We opted to build one. It's not the most flashy greenhouse. It's at the lower right hand, and that was built in 2016. So there are some assumptions that we use on our crop production at MDRS. And a couple things to remember. We're an operational analog, so we're not looking at high tech here, okay? We don't want a lot of bells and whistles in our system, simply because it's super expensive and it's not really something that we do. We have been using traditional methods of growing things in soil because our assumption is that the soil issues on Mars will be worked out before we get there. And so we'll be able to modify Martian regolith and whatever else we need to grow crops there. We've done both aquaponics and hydroponics at MDRS. It's far too labor intensive for us to want to take it on. When you put those systems out in a field station type setting, and then you ask different people to learn how to take care of it on the fly, it just doesn't work. Our crop production here at MDRS is only for supplemental food. We are not growing enough food to keep the crews fed. And our model is for a Mars station the size of MDRS, so we're not looking at a settlement of 100 or 1,000 or 10,000. We're looking at, under a dozen people. Okay, so the first thing we learned when we got this new greenhab is that crews don't want to plant, crews don't want to harvest. They do like spending time in the greenhab, and they do enjoy having the supplemental food, but the actual taking care of it is too big a challenge. Part of that is that we have some really dedicated, amazing greenhab officers, but a lot of the crews, that's kind of like everybody picks a role, and then there's one last person, and they're like, okay, well, I guess the only thing left is the greenhab, and so I'll do that. And that doesn't bode well for crop production. A great example of that is the crew that just left. Inga, who was the greenhab officer, had a lot of farming experience, and she actually harvested in more material in the 2 weeks that her crew was here than basically any other crew. And there's hardly any crops in the greenhouse right now. They're all just young because it's just the beginning of the season. So that was one of the things we learned is, you know, I was expecting the crews to passionately want to grow their own crops, and nobody did. In 2017, I decided I was going to start all the crops, and we had a bumper year, and we have ever since. So I learned a valuable lesson. It's not that they didn't want to plant, and it's not that they didn't want to harvest. It's just that they really didn't know where to start. And so prior to the field season beginning, we plant the greenhouse completely. And then the crews, as they come in, they kind of have to maintain the crops. And I guess everybody is relatively good at keeping things alive, but they might not utilize the greenhouse in a way that they should. So then we decided to look at crop production over 2 field seasons. In 2018, 2019, we had a field season, 216 days, and we got almost 5,000 grams of harvested food from the greenhouse. About half of that was crops. That would be tomatoes, peppers, cucumbers. The herbs were about 20% of the harvested biomass, which is kind of interesting because it stayed that way even the next year. And the greens were about 36%. The daily harvest over that field season was 22 grams a day. The next year, and remember, this is a COVID year, so we only had 132 days in that field season. We harvested almost 4,000, which is an improvement. We're seeing about the same percentages, but we're getting about 30 grams of food for the crews a day. And that year we would have seen a much higher result, because we would have, if we had gone all the way through the year and not had to interrupt the field season because of COVID, because I had to give away all the plants that were in the greenhouse and they were just starting to produce. So 2019, 2020, we would have got ahead of, we would have had a really good year if we had kept going. So what we need to do, we need to select and or train our greenhouse officers so that they're more competent and more comfortable in the greenhouse. So that is one of the things that we're working toward. We have to record, we're going to record the harvest data just as we always have. And we're going to try to increase the harvest to justify the cost in time, energy, and resources, and especially water, that we're only seeing this small amount of food come out of the greenhouse. We're going to track those costs beginning with the Purdue crew in December, because from December on, we should have a full field season and no more travel restrictions to interrupt us. And then with the help of the 2 interns that we have coming this season, we're going to design a crop production plan that reflects what people think we're going to need on Mars. So we're going to balance what we have, what we expect on Mars, and come up with something that is practical that we can do that reflects current thinking of what's going to happen on Mars. And then we'll implement that design in a complete experiment in the next field season, so in 2022, 2023. We welcome anybody who wants to work on this project. I guess my one request is please don't try to sell me on hydroponics and aquaponics. We want us to kind of support the assumptions that we've already made. And that's it. So I'm going to turn this over to the next speaker, who I've totally forgotten who it is. Who's next?
Okay, yeah, thank you so much. My name is Atila Meszaros. I'm the Assistant Director of MDRS, and I'm the lead of the biology project that we're going to do at MDRS in the next few months this season. Shannon said, we've been working on this project for a while, and we are really excited to to finally be able to execute it. So, well, I'm gonna talk tonight just a little bit, like just an overview of what we're planning to do the next few months. So, as you probably guess, project is going to be related to cyanobacteria and also lichens, but most importantly, it's going to be about the geomicrobiology at MDRS. We want to focus on the interactions, not only between the bacteria, the microbe community, the surroundings, but also how they interact with the geology of the place. So in order to start, I'd like to talk a little bit about what desert varnish is. I don't know if many of you have heard it before. Actually, it was my first time to learn about it after we start this new approach of the project. So desert varnish, sorry, is this dark coating in the rock, in the surface of some rocks in arid and semi-arid environments all around the world. And some of the characteristics that it has is that it's very rich in clay minerals, iron oxides, but most importantly, I think the most defining characteristic it's very, very abundant amount of manganese. And one of the interesting things that we also want to understand is that why does it have so much manganese? It's a question that is still unknown, but most importantly, we don't know yet how desert varnish is formed. So we don't know if it's a abiotic process or a biotic process. So that's one of the questions that we want to answer, because we know, well, not answer, but we're gonna go around that's one of the big questions that we're gonna go around. And so we know that desert varnish shares a very complex bond with the microbe community. And one of the big questions that we have is the desert varnish biologically mediated? Also, we want to know which factors are going to determine the community compositions. So we have, I mean, like everywhere, we have, and sometimes we don't think that desert places will have such a rich communities, but they do. And we have the desert varnish community and the lichen community that they constantly interact between each other. So one of the questions that we have is how much they compete between them, how much they collaborate, which ones are the first to get there, and how they all interact with the desert varnish it's one of the main concerns that we have. Also, what about its distribution? Why they're laid out in this position specifically? What are the benefits? Why are they behaving this way? Is basically what we're trying to figure out. And finally, well, not finally, but because we have a lot actually of questions that we're still working on. But we want to know if the desert varnish can be a reliable biosignature for future mission on Mars. We know there's a lot of rocks coating in Mars that share a lot of similarities with desert varnish. And this, if we somehow figure out how desert varnish is formed, and it is indeed biologically mediated, this could be a great opportunity for the use of desert varnish as a reliable biosignature. And that's pretty much it. We still are working on a lot of our hypotheses and our methodology, but we're really excited about this project. And hopefully our timeline is going to be, we're going to start in December, hopefully wrap it up around February, March, and looking forward to present it on the next Mars Society convention. I think that's pretty much it.
My name is Jen Carver Hunter. I am a fifth grade teacher in the Salt Lake City School District, and I have been on both of the Spaceward Bound Utah crews that we've had so far, and I'll be coming back as the crew commander for the third crew in a few weeks to work with Shannon and Atila on that. So I'm going to talk a little bit about the Spaceward Bound Utah program. Can you, Carrie, can you see my presentation okay?
You look great and you sound great. Go ahead.
Perfect. So the Spaceward Bound program is from a 5-year NASA grant that was awarded to Dr. Shannon Rupert of the Mars Society. And it's designed to train the next generation of space explorers by having educators participate in analog experiences. And to date, we've had educators from third grade up through high school, as well as informal educators who have participated in this experience. Spaceward Bound Utah began in 2019 as a means of providing a Mars analog experience for Utah teachers. And while most of the crews that go through MDRS have 2 objectives of the sim and the science, with the Spaceward Bound Utah program, we do add a 3rd objective, which is education. So we really have balanced our time in this experience with not only having the sim experience and the science exposure, but also increasing our content knowledge as well as our professional knowledge around the field of education. So sim pictures, of course, look very familiar, I think, regardless of what crew is on the sim. But we also do have different science things that we do. We've had collaborations on science investigations that we can take back to our classroom and differentiate from everything from an elementary level up to a high school level. We have analyzed specimens. We have searched for extremophiles. We've also conducted geology EVAs where we collect samples and to identify and use as analogs for what we are going to be talking about through the experience at the MDRS. In addition, we've done investigations with bacterial collection and analysis. And finally, the most important piece for educators that's different than other crews is the education piece. This experience gives us designated time for collaboration. So our hats never come off. If we're not doing science or actively on an EVA, we are collaborating. We are conducting professional development and exploring resources as well as any suggestions other people might have.
Jen,
we leave the
Jen,
sorry to interrupt you, but you're
we're both talking, sorry.
Sorry, you go, Gary.
Your presentation started to go and then it stuck. So now it's back on the first slide.
Oh, let's see. There we go. Is that better?
It's on the education slide. Is that the slide you want to be on? Okay.
Yes, that's where I want to be. Thank you for that.
Thank you.
As we are part of this program as educators, we lead with resources. We lead with a greater professional network of teachers who are passionate about the same depth of education that we all are, and we leave to go back to our students and develop a love for Mars in our students so that we can hopefully encourage the next generation of space explorers.
Perfect. All right. Well, thank you very much, everyone. My name is Paul Sokoloff, and I'm a botanist at the Canadian Museum of Nature in Ottawa, Ontario, Canada, where I live and work on Algonquin and Anishinaabe land. And today I'm going to talk to you a little bit about the research that we're doing in terms of biodiversity of the analogs around MDRS. So, you know, when we think of the Mars as a research station, we think of it as this operational analog, which is amazing. And we've had so many wonderful crews go through and figure out, okay, how can we live and work on Mars? But we also have the broader environment, which is situated around MDRS that is incredibly biologically rich, as you've probably gleaned from Shannon and Atila and Jen. And so, you know, I'm going to talk to you a little bit about what we do botanically there for our biodiversity research. Of course, I couldn't start off a Martian botany talk without a quote from Mark Watney, which I've modified a little bit here. I think the original quote is "Mars will come to fear my botany powers," but ours is appropriate because this is the work of an amazing group of people. So the Martian biology program is the brainchild of Dr. Rupert, myself, David Murray, Samantha Macbeth, Mike Irvin, Jeff Zarla, all these amazing people who have contributed over the years. So we all know what a botanist is, right? Mark Watney is a botanist. Well, we're not those kind of botanists. We're not growing things in metabolic waste products, fortunately. No, we're the kind of botanists who are actually interested in what kind of plants are in the environment around us. How can we tell them apart, and how can we communicate that information to other folks? So what we use is, instead of potatoes stored away for Thanksgiving, we use things like a quadrat and a plant press to essentially, and then our own intuition about environments, and as Shannon mentioned, wandering. We go out into the landscape and we are essentially search out all of the different microhabitats that surround MDRS and make collections of the plants, the lichens, the algae, and the mosses that grow in that area, so that we can preserve these specimens and examine them to identify them to species. And so what we do essentially is we go out, we collect a plant, we bring it back to the Science Dome, we put it in a plant press, which you can see an example of it here, you know, state-of-the-art technology circa like the 1800s. And then once the specimen is flattened, essentially between sheets of cardboard and dried out, we mount it on sheets of acid-free paper with a label that says where we collected it and when we collected it. We deposit a set of these at the Intermountain Herbarium in Utah, and also a set here at the Canadian Museum of Nature. And within those libraries of dried plants, we essentially have a single specimen represents a data point saying that this particular species was collected at this place and at this time. So we have physical proof that these species are found in and around MDRS. So as Dr. Rupert mentioned, we started this program back in 2017, and we actually did some collecting before that in 2014 as a part of the initial trial run with Crew 143 for what was then the Mars Arctic 365 mission, and which turned into the incredibly successful Mars 160 mission. So I have done sim collecting of plants in a simulated spacesuit, as you can see here. It's not the greatest. It's actually a little bit better to do it out of sim, which is why we've been doing these Martian biology programs as non-crew projects. And so we've been going as much as we could. We were hoping to go last year, but COVID. And with any luck, next year we'll be back in the deserts of Utah, figuring out what kind of plants and other organisms are there to contribute to this bigger mission profile that we're working on. So in terms of vascular plants, and these are things that are mosses, so trees, things that have roots and shoots, we have found to date 81 taxa within the operational area around MDRS. And I say taxa, not species, because some of these things we actually haven't identified at the species level. In particular, there are some endangered cacti that are in and around the Mars Desert Research Station that are federally listed, so we don't collect them, but we do examine them in situ, and we come to at least the genus that they are. But there's an incredible amount of diversity in vascular plants surrounding the station, and in an immense array of microhabitats that provide shelter for different groups of organisms to grow together. And I should mention too that there are high levels of endemism in the area south of the San Rafael swell where MDRS is located. Endemism means these organisms are only found there and are found nowhere else on the planet. So, you know, this is an incredibly rich area to be conducting field botany programs. And so, you know, as we were talking about wandering, it's just exciting. All right. We're also working on lichens, which will be really useful, we know, not only to Atila's project, to many other research programs that are looking at microbiology and geomicrobiology around the station. To date, we have 13 species of lichen recorded from around MDRS, You can see some of them here. And this one in the middle, actually, this white cracked one is called, in the Latin name is Acrospora strigata. And I forget what the English name is at the moment, but it is the #1 most common lichen species around MDRS. It's everywhere, it's abundant, and having that identified as species makes it a good model organism for future studies. We have 13 species so far, but we're relatively certain that's going to go up thanks to the amazing efforts of the Mars Desert 80 team, including astrobiologist Anusha Sharma, who is here, who has collected a lot of lichens, and who I'm working, these collections that I'm currently working on with colleagues at the museum to identify. Lichens are interesting because unlike vascular plants where we have just to look at them physically to key them out, lichens actually require specialized identification techniques, things like TLC, thin layer chromatography, and also chemical analysis. And so going through that actually provides some interesting science return in terms of how do you do science in the field. We've worked a little bit on the biodiversity of microbial green algae and cyanobacteria that occur endolithically around the station. We've identified 6 taxa, and that was all done morphologically. So all done by looking at the species under a microscope and identifying them based on visible characteristics. This shows a bit of a limitation in that I'm certain that if we did a genetic study, we would get a higher level of, okay, these are the number of taxa that are here. But that provides an interesting contrast for old ways of doing things and new ways of doing things. And we'll need multiple tool sets when we're examining both, searching for life on Mars, but examining life here on Earth. And so after all of that, you may be asking, okay, what does this have to do about Mars? There are no plants on Mars. We're looking for life, but it certainly won't be anything that you can stuff inside a plant press. And the answer to that is twofold. The first being that we have at MDRS this amazing analog area. And by better understanding the biodiversity and the botany and the lichenology of this area, we can better characterize it as an analog. By knowing what's there, astrobiology crews and crews going to the station have a baseline to work from for their own experiments. But the second reason, and one that motivates a lot of us, is that we all really want to see crewed exploration of Mars. We really want to see people go there, but we also know that we have a pretty amazing planet here too. And at the end of the day, once we do hope that we'll eventually have people on Mars, we'll want to have this amazing place for them to come back to. And by better understanding the environment of this really unique desert, we're better able to do things like understand how endangered species live in this area, understand invasive species and how they spread in the area. And that gives people like folks at parks and land managers at BLM the ability to better understand the area and give back a little science in this place that so many of us have come to know and really love. So that's in a nutshell what we're doing with the group. Our future work is to hopefully go back this coming year and expand this beyond the flora and look at some of the fauna, things like insects, arachnids, and do some mammal and bird surveys in collaboration with other scientists in the area. And of course, we always have, Dr. Rupert's wanders around the station sending us cool pictures of plants. So we're always so keen to get back there. If you're interested to read up a little bit more, you can find our current work published in the Biodiversity Data Journal. And I think that might be the end of all of us speaking. And we have time for questions.
Thank you so much, Paul.
Yes.
The work that you guys are doing out there is absolutely incredible. Shannon, did you have something to add?
No, I guess, aren't I lucky to work with such amazing people?
Absolutely.
We're lucky to work with you.
It's not easy being out
Thank
at
you, Paul.
MDRS like Dr. Rupert is. So kudos to her for sure. The incredible work that she is doing with all of the crews that she has come out there to work together. It is not easy. So thank you very much, Shannon, for everything that you do out there. And Attila and Jen and Paul, thank you very much. Let me see if there are any questions for any of you. If you are in the audience and you do have a question for them, please feel free to put your question in the Q&A section. I did have one question. Attila did answer, but maybe you could give some more specifics on this question. What is the area of your greenhouse? Perhaps what it's made of, how it's structured, etc.
Oh, so the greenhouse is 12 feet by 24 feet in size. It is a, if you want to know a secret, it's not actually a greenhouse, it's a single car garage, because we were unable to get the greenhouse that we ordered. And so it's a single car garage kit that's been covered in greenhouse material, as opposed to just, you know, standard metal. And it has heating and cooling. We have grow lights for when we start seeing some flowering. I increase the light because we grow from October through about April. And of course, in the winter, you know, I always like it when I first start growing in the fall, because the seedlings are like, what the heck, why, wrong time of year, lady. And it always just kind of cracks me up. Is there any specific questions about the greenhouse?
I'm just going through the questions now. And one question was, is there any particular time that you need help with the greenhouse, or is that even an option?
Yeah, I mean, we start planning in September or October. September, it's really too hot until the very end. And then it gets hot. It's too hot at about, we shut down at the end of April. So that's our active growing season. But we'd like to have, you know, brains on board all year round that have, you know, to work on this project with us.
Wonderful. We're going to go out to the AltSpace VR. Can you hear me?
Yeah, thank you, thank you. This would be, I think, for Attila or Paul, and your work with the lichen. It sounds like you might be looking into the patterns of them competing with each other. And I'm curious, have you found any that you maybe can find analogs that you can model with the Turing patterns? You're familiar with those, the mathematical patterns that show things like zebra stripes and bacteria colonies and things, if you're familiar with that concept, Turing patterns?
Actually, no, that's my first time hearing of that. But you mentioned competition, and certainly there are aspects of competition within some of the lichen species around MDRS, but there's also parasitism amongst different lichen species too. In fact, some of the lichens are obligate parasites on others. So, and there's also, you know, a lot of, as Attila and Shannon can speak to, desert soil crust communities that are interwoven that maybe follow those patterns.
Interesting, interesting. Yeah, it's something I recently discovered too. Alan Turing, the guy who worked on our code breaking and those in World War II, he also did some work in biology and understanding some of that. So it was something I recently discovered. Turing patterns. Look into that. It'd be kind of cool. Thank you.
Thank you so much.
Cool. Thank you.
I have to say the AltSpace VR interaction during this convention this year is incredible. Our next question comes from Peter. And I know that you briefly touched about this, but maybe you could go into a little more depth. What forms of heating and cooling systems do you have for the greenhouse?
Okay, so the greenhouse is heated in the winter by a greenhouse, a traditional greenhouse heater. It's propane. It's temperature controlled. For cooling, we have a swamp cooler, basically, and it's not effective all of the time. We also use a lot of different shade cloths because even now, the temperature can get pretty warm during the day. Right now I have a 90% sunshade on, and probably in another couple of weeks we'll switch it out to 40 or 50. But we're definitely heating every night now. And but we have, the greenhouse is filled with seedlings and little guys that are only 4 or 6 inches tall.
Thank you, Shannon, very much. I have one more question. And it's from Eric, and he asks, what are the most promising crops to produce food oils? And he was wondering if sesame is at the top.
That's a good question. I don't know. I mean, when we started growing food in the greenhouse, it was relatively easy for me because I worked in greenhouses my whole life. My family were organic farmers, and then I did a lot of work at university in greenhouses. But I hadn't given a lot of thought to, what exactly would be required on a Mars mission, certainly early on. And then it was funny, the first few years, it was like whatever seeds I could find at the end of the season. And now I remember to buy the seeds in the summer, and I know exactly what I want to buy. But we're not, I mean, we're very slowly stepping into things like medicinal plants. The first thought was like, oh, let's grow greens and let's grow tomatoes. I love cherry tomatoes. And so we haven't really thought beyond what would be, kind of plants that would make the crews feel like they were getting a gift out of the greenhouse. So I don't know. That's a really good question, and it's certainly one of the ones that we'll want to ask. I listened to a talk earlier today, and they were talking about all the starches, and all the the wheat and the corn and the cassava and all of these things. And I thought that's really interesting because we don't actually grow a lot of those. Part of them is the practicality of having a 9-month field season, and the other part is just that it was such a struggle just to get crops to grow, and you would not have believed how hard it was to actually maintain a greenhouse here for the time that we do. And it's not necessarily that because we don't have a lot of technology, and so people will argue, well, yeah, sure, because you don't have this really sophisticated setup, and that might be part of it. Part of it too is everything you do on Mars is going to be hard, so much harder. If you'd have told me that 5 years after actively starting a greenhouse program that we would only be this far along, and part of that is just being busy and having other things, then it's more important to just get food that they can grow. But also everything out here is really hard. It's a crew can expect to do about half of what they think they can do, even if they have reasonable expectations. Everything is just a lot harder. And so that's a great question. I'm looking forward to actually exploring what would be what would be the best crops instead of what is the easiest and quickest growing, so that all crews can have a little bit, the idea is that at least if you're growing a lot of greens, and we grow a lot of greens, then every crew gets to at least experience harvesting. So, that is a great question. And now I want to know.
Okay, thank you, Shannon. One more question. What is something very unusual that the crews have made from something that they've harvested there, but was that they all enjoyed, but was highly unusual?
Unusual. I will tell you that the not unusual thing is pizza. I mean, I think almost all the herbs that are ever harvested are for pizza. Let's see, I can't think. Oh, I know what the most unusual thing is we were growing zucchini and I got a message asking if they could pick the zucchini flowers and eat them. Oh. and so if you actually look at the data, it shows that like we harvested 4 grams of zucchini and you're like, how can that even be a zucchini? Well, it was just because it was zucchini flowers. So, and apparently wherever the crew was from, I can't remember, it's a very common thing for them to eat. And so they wanted to eat that.
That's amazing. We do have time for a couple more questions. Cool. I have one coming right now. George asks, since crops will be grown in a greenhouse on Mars, and the growing times are fixed seasons like on Earth, or is there a way to possibly grow crops year round?
Oh yeah, we'll be able to grow crops year round. I mean, the one thing about traditional agricultural methods is that we know how to, trick plants into flowering when they don't want to, or like I said, these little babies, they pop up and they're like, whoa, where am I and why am I growing at this time of year? And so we'll be able to have year-round crops, and even in the time that we're here, we turn over quite a lot of herb and greens. So even though for our crops, those things like onions and tomatoes and zucchini and cucumbers and peppers, we only get one crop during the field season. The other things are constantly turning over, and so we could definitely grow year round. And if you think about it, if you followed agroecology practices, which we sort of do on a very, very minimal scale. If you notice the picture of the plants growing, our herb garden is actually a recycled, reused aquaponics bed. And we grow carrots in an aquaponics bed too, because it's deep. It's the one that have the fish in it. And so, you can have a schedule that allows you to take advantage of rotating your crops even in a tiny environment like what we have.
Thank you, Shannon.
Although sometimes it is a challenge. Sometimes it's a bit of a challenge to figure out how much light everybody should get when somebody's doing one thing and some other plants are doing something else. And so it's a bit of a balance, trying to figure out how to keep everybody happy.
I'm sure. I just try to grow my garden here in Colorado, and I don't have the situations that you have at MDRS, and I have a hard time here. So your crews and you must do an amazing job out there to grow things, because that environment is not very friendly sometimes. I do have to say, I have a comment from Tom. He said his wife loves zucchini flowers in Italy. So that's a real thing.
Oh, okay. Cool. All right. Okay.
Does anyone else have any other questions? I think I have one more question. Peter asks, have you tried growing leeks? They have a long life in Brazil.
No, we haven't. And I'm not really sure why. I mean, I grow things somewhat related to leeks, you know, green onions, and I love leeks. I'll look into that. That would be a good one. That would make me happy. So I'll check into it.
Well, we do have about 5 more minutes. I don't have any more questions at this time, but Shannon, Atila, Jen, or Paul, if you have anything that you would like to add to the end of your panel, please feel free to at this time. We have about 5 minutes.
Well, I'd like to bring up the fact that we're very excited to welcome our first interns in our new intern program this field season. We have, it was really funny. I guess I'm so used to putting crews together that when it came time to select a single intern, instead I saw these amazing teams of interns, and I don't know where they're all going to sleep because I got a little over exuberant. But we have three interns coming to work on the astrobiology desert varnish project. One is a grad student who's actually worked with Penny Boston, who's on this project with us in the past. And then we have 2 other, one is a recent graduate, and our third intern is a early college, first in her family to go to school person. And they just, together, they made this amazing team, and none of them had any field experience. And I'm like, oh my God, this is gonna be so cool. We're gonna bring them out. We're gonna have them doing all this field work. It's just gonna be mind blowing. And then for the agroecology program, which is the green hab work, 2 of the applicants, one was very interested in crops and genetics, and the other one was all about the technology and the management of the system. And together, they just made like this perfect team. And so I ended up inviting them all. They're all going to be here through probably mid-December to mid-January. And even though we've remodeled the intern trailer, we still don't have enough beds for everybody. So, but I have a couple months to figure that out. I'm super excited to welcome all 5 of them. And, you know, it's that we have more interns this season than we've ever had. And all the other times that we've, Atila being the first intern that's come, we've only had 2. So it's going to be very exciting around here this winter.
Yeah, and I just want to say that I'm very excited to see. I mean, I was the first intern not that long ago, and then we had David and Lauren, and I'm just very happy to see the program keeps growing with the time, and just we have more people on board on our ship.
Yes, I'm sorry, I forgot Lauren. Lauren actually never made it out to MDRS. I hope she gets out of here sometime. She was our intern during COVID. So, and she has remarkable organization skills. I wish I had some of those. You know, I learned a lot from her. The other thing I want to mention is that we do have cohort 3 of the NASA Spaceward Bound program at the end of this month. And I'm really excited before they come, we're having a reunion of cohorts 1 and 2. So these teachers, they just blow me away. Jen is an amazing, amazing person, and she's made it, her enthusiasm and her wonder at everything that we have here has just made her the perfect commander, because she's so inspiring. And I'm so excited that they want to come back, and we're going to put together these kits that are part of the program. We decided very early on that we didn't want to make curriculum or that there was so much curriculum and so much information online that we really wanted to put physical things in the hands of other teachers. So we've developed these kits that contain books and activities, and those will go to Utah teachers, but mostly middle grades that aren't able to come to the program for one reason or another. And so they're going to come, we're going to put those together, and we're going to have a Halloween party. I'm so excited about that, even though I hate Halloween parties. And so it's just been an amazing program. I'm really proud of it, and I'm really impressed by it. I mean, I know it's my program, but I'm still impressed by it.
Well, you should be. There is amazing work being done at MDRS. So this is going to conclude this first hour of this evening. Dr. Shannon Rupert, Director of MDRS, Mars Desert Research Station, thank you very much. Thank you, Atila. Thank you, Jen. Thank you, Paul, very much for joining us. Continue to do your good work out there. It's absolutely amazing. Our next.
Thank you, Carrie.
You're very welcome. Thank you all.